Good morning or afternoon or evening, and welcome to this talk at the ICAP 2021 conference. My name is Lukas Orre, and I'm a senior researcher at Karolinska Institutet with a research focus in MS-based proteomics, method development, and application in cancer research. Let me just share my slides. Today I will tell you about a recent study describing the proteome landscape of non-small cell lung cancer. Lung cancer is the most common cancer type, as well as the deadliest, causing more than 2 million deaths worldwide each year. Lung cancer is divided into small cell lung cancer and non-small cell lung cancer that is further subdivided into adenocarcinoma, squamous cell carcinoma, large cell lung cancer, and large cell neuroendocrine cancer. Overall 5-year survival is poor, below 20%, mostly due to that lung cancer is typically detected at late stage when the cancer has already spread to distant locations. Luckily, a growing number of different treatment options are now available also for late stage lung cancer. In addition to systemic treatment with chemotherapy, targeted therapies against ALK, EGFR, ROS1, and BRAF, as well as immunotherapy can now prolong survival several years, even in advanced cases. Importantly, these therapies are only effective in subsets of patients, and we have very little knowledge about how to combine these medicines for optimal effect. To elucidate phenotype-level subtypes of non-small cell lung cancer, we used a cohort of surgical samples representing all major histologies of non-small cell lung cancer. For reference, we also added 2 small cell lung cancer samples. Protein extraction was followed by digestion and TMT labeling for relative quantification of proteins across samples. Pre-fractionation before LC-MS for improved analytical depth was performed using our own high-reef method. This setup resulted in the identification and quantification of close to 14,000 proteins, with almost 10,000 proteins quantified across all 441 samples. In addition to protein level analysis, we also had access to panel sequencing data covering 370 genes and chip-based transcriptomics and methylation data for a subset of the cohort samples. The high-reef method was developed specifically for in-depth proteogenomics analysis, and the fractionation relies on separation of peptides based on isoelectric point using immobilized pH gradient strips. Due to extremely sharp separation, each generated fraction contains mostly unique peptides, resulting in very efficient reduction of the sample complexity before LC-MS analysis. The overlapping close to 10,000 proteins in the analysis were used for consensus clustering of the 141 samples in the cohort, resulting in 6 distinct clusters or proteome subtypes. This clustering was driven, as expected, by histology, with subtypes 1 to 4 enriched in adenocarcinoma samples, subtype 5 enriched in large cell neuroendocrine cancer, and subtype 6 enriched in squamous cell carcinoma. Importantly, subtypes were also driven by oncogenic signaling, where, for example, subtype 1 was enriched for EGFR mutations and subtype 4 enriched for STK11 mutations. Further immune infiltration was characteristic for subtype 2 and 3, but with different immune types, cell types. Importantly, proteome subtypes were distinct from mRNA-level subtypes, underlining that proteomics analysis contribute novel information. Next, we performed network analysis of proteins that were significantly different between subtypes. This analysis revealed subtype differences based on both signaling and cell type. As example, subtype 1 showed strong expression in module describing alveolar epithelial cells, while subtype 5 showed strong expression in proliferation modules. Focusing on proteins previously shown to be implicated in cancer, our data indicated common overexpression of known oncogenic drivers such as EGFR, ERBB2, and KRAS, but also oncogenes previously not implicated in lung cancer, such as the kinase SGK1. Oncogenes were more commonly overexpressed in more highly proliferating subtypes, such as subtype 5. Analysis of the regulation level by evaluation of correlation between promoter methylation and mRNA, or between mRNA levels and protein levels, indicated widespread post-transcriptional regulation, such as microRNA, translation or degradation-dependent regulation of protein levels. Further analysis of the immune landscape of lung cancer showed clear infiltration of immune cells in subtype 2 and 3, but with more T-cell infiltration and interferon gamma signaling in subtype 2 and more B-cell infiltration in subtype 3. Importantly, subtype 2 samples showed high tumor mutation burden, as well as antigen presentation, indicating that neoantigens recognizable by the immune cells were produced as well as presented on the surface of cancer cells. Our analysis further indicated that subtype 2 cancers used overexpression of PD-L1, a ligand to the inhibitor PD-1 receptor on T cells to evade killing by the immune system. Mutations are not the only source of neoantigens in cancer, and for a more comprehensive analysis, we went on to investigate the levels of both cancer testis antigens and non-canonical peptides in the lung cancer samples. These proteins are not supposed to be expressed in lung cells and can therefore trigger immune cell-dependent killing. Interestingly, both cancer testis antigens and non-canonical proteins were more commonly produced in subtypes 4 to 6, resulting in higher tumor neoantigen burden. Further, our analysis suggested that reduced DNA methylation and looser transcriptional control in these subtypes was responsible for high neoantigen levels. The finding of higher neoantigen burden in immune cold subtypes indicates specific immune evasion mechanisms. A closer inspection of known ligands to the inhibitory T-cell receptors indicated, as already shown, high levels of PD-L1 in subtype 2. Importantly, 2 of the subtypes with high neoantigen burden showed high levels of other checkpoint proteins, B7H4 in subtype 6 and FGL1 in subtype 4, suggesting new subtype-specific immune evasion mechanisms. Both these ligands have been shown secreted, suggesting that they may be involved in systemic inhibition of the immune system. FGL1 is a protein normally expressed in liver cells, and to investigate the high FGL1 levels in subtype 4 further, we performed a protein-wide correlation analysis to find other proteins possibly linked to FGL1 regulation. This analysis indicated 2 additional proteins with liver-specific expression, the transcription factor HNF1A and the metabolic enzyme CPS1. Interestingly, FGL1 also showed a strong negative correlation to the tumor suppressor STK11, but only at the protein level. Further analysis showed a very strong connection between inactivating STK11 mutations and high FGL1 levels in our cohort, as well as in public domain data. STK11 signals through AMPK, and we could show that treatment of the STK11 mutant cell lines with an AMPK activator resulted in reduced levels of both HNF1A and FGL1. Based on this, further a rescue experiment, we could show that re-expression of wild-type STK11 in lung cancer cell lines resulted in loss of HNF1A and FGL1. Based on this, we could conclude that inactivation of the tumor suppressor STK11 in lung cancer has the potential of both increasing metabolic activity and proliferation and driving immune evasion through increasing the levels of the immune checkpoint ligand FGL1. Our analysis thus indicated that the subtypes identified could provide important information related to oncogenic drivers and immune evasion mechanisms with promising implications in therapy selection. Therefore, we developed 2 classifiers for subtyping lung cancer samples in a future clinical setting. For classification of sample cohorts, we developed a support vector machine-based classifier based on the original MS data. For classification of individual label-free samples, we first reanalyzed the non-small cell lung cancer cohort using DIA-MS and developed a K-top scoring pairs-based classifier using this data. Both classifiers showed high accuracy in classification with very limited false classification rate, or low. For evaluation of our identified subtypes and developed classifiers in public domain data, we used a metadata set with mRNA levels as proxy for protein levels. Classification of this large cohort containing 925 non-small cell lung cancer samples reproduced with close to identical characteristics the 6 proteome subtypes in terms of histology, marker expression, and infiltration patterns. Importantly, the size of this cohort allowed for evaluation of survival, indicating that the identified subtypes included important prognostic information as well. For additional validation of the developed DIA-MS-based classification pipeline, we analyzed an independent early-stage non-small cell lung cancer cohort of more than 200 samples. 175 samples with sufficient coverage of classifier features were successfully classified with the single sample classifier, again demonstrated expected subtype characteristics. The majority of non-small cell lung cancer cases are diagnosed at late stage when surgery is not an option. Consequently, no surgical material is available for this analysis, only minute amounts of biopsy material. To investigate if our classification pipeline was sensitive enough also for this type of material, we performed DIA analysis of additional cohort consisting of 84 late-stage non-small cell lung cancer biopsy samples. Even though the number of identified proteins per sample was lower than in the validation cohort due to limited sample amounts, our analysis showed promising results with classification of 58 late-stage samples with the expected histology distribution between subtypes. In summary, our analysis indicates 6 distinct proteome subtypes of non-small cell lung cancer. These subtypes were defined by growth pattern, cell types, oncogenic drivers, immune phenotypes. We could also show prognostic value and targetable subtype-specific immune evasion mechanisms, indicating a future clinical diagnostic implication of these proteome subtypes. For future diagnostic applications, we developed a DIA-based classification pipeline and demonstrated the applicability for subtyping of non-small cell lung cancer in early and late-stage cancer. This data has been accepted for publication in Nature Cancer, so you can read the full story within short. I would also like to acknowledge all the people that have been involved. This is the cancer proteomics groups headed by Professor Janne Lehtie. And specifically, I would like to acknowledge the students, Tanner, Yanis, Fabio, Jambu, and Olena for their hard work in this project. As collaborators, we have a group in Lund, headed by Johan Staff and Maria Plank, an Oslo group, and a group in Edinburgh. With that, I would like to thank you all for listening, and here are my contact details as well, and hope to see you live next year at ICAP 2022. Thank you.